Sorry.
Yeah.
So now we're going to go on to Weka itself. Obviously, just for those who are watching and are not in the room, I've just been talking around the package manager for Weka, referring to deep learning packages.
So it would be much better if you could just put it on your shirt.
Okay, sure. Okay, is that better?
Yeah, thank you.
Okay. So I've just been talking about deep learning for J. I've just been talking about AutoWeka as being useful tools to install. So there are various packages or applications within Weka, and they implement machine learning in different ways. So the first one, and the most easy to use and the most commonly used one, is Weka Explorer. And this is a GUI, so it's a graphic user interface. It means you can put things in place, implement solutions, and you don't need to do any coding. So what I'm going to do, first of all, I need to load a data set in. Some important things to know. I'm just going to go to my PC, and I just want to, right.
Okay. So just switch it off. That's why the sound is on.
Shall I try one of these?
Maybe you can try. Just turn on.
I'll use that one. A bit. I'm just going to show you the format of a file. And just something particular to Weka that you have to do. Oh, this is awful. Maybe you don't need the volume down or something. Is that better? The key thing to note here is where we have a class in Weka, we have to use a word to denote the class rather than a number. So normally we would use binary notation. 0 is one class, 1 is another. Here we use a word. So pos means disease positive, neg means disease negative. That's the only real difference. Everything else in Weka can be loaded as a CSV in the same way. So I need to load some data. So first thing I do is open a file. Let's go up to the relevant directory. Negative, blue is positive. So essentially it's providing me histograms just so I can see what my data looks like. So that's the file load interface. I can choose filters for that. So I can use smoothing filters. Say we're using mass spec raw signal, you could smooth those signals out here with filters. So I can load those filters in. I'm not going to use them now, but there's lots of ways of filtering data in Weka. There's not time to go into those right now. So what I'm also going to do, first off, is I'm going to select all the variables, and they're the ones I want to use. So by selecting those tick boxes, it means I'm going to use these for the analysis. Right, so now I move from the pre-processing tab up the top here to a classify tab. Now, again, I can choose one of many classifiers. You see here I have Bayes classifiers, I have functions, so I've got a multi-layer perceptron here, I've got deep learning as a classifier if I want to use deep learning, simple logistic regression, many other things. I can also use rule-based clustering, 0R and part, and those, many other types as well. And you can see here I've got decision stump, I've got random forest, I've got random tree, I've got J48, which is another classifier based in Java. Nice thing is, every time I move over one of these, it tells me what it is and how the data should be formatted for that. So I'm going to start off, I'm going to make a random forest. So now I've got this random forest. If you look here, this is the parameters, the hyperparameters for that random forest. Stop it. So if I click on this, I can immediately start changing the hyperparameters in this. If I look at the help for Weka, it's not immediately here, but if you look it up in the help, say random forest, it will tell you what these mean. But if you move the cursor over, it does give you an indication as well. So for example, number of iterations, how many trees am I going to make in the forest? If I look at number of decimal places, that's just rounding. If I look at the batch size, this is the number of instances to process. I can print the classifiers, I can do various other things. I can also save this if I want to. So if I've got a particular form of hyperparameters that work, I can save them here, or I can open new ones. But I'm just going to click OK. What I also have here is the testing options. So I can use a cross-validation, which is like a bootstrapping. I can use 10-fold cross-validation, so 10 different splits of the data. I can also use a percentage split. So if I want to train on 66% of the data and use 33% as testing data, that's where I can do it. I need to set, I can have other options, there's a whole load of other stuff I can change here. I need to set the class that I'm predicting. Well, if you were asleep, that's certainly woken you up. So, I'm basically selecting the output class here, and I've set it all up. So what I now do is I click Start, and you can see it's quite quick. It's a small data set, but I've got immediately various things. So I've got how the classifier has been set up. I have the number of iterations, things like that. Then I have the correctly and the incorrect. What? Dear me. What is going on with this machine? So I've got the correct and incorrect classes. I've got the error, and I've got the true positive rate, false positive rate, the rock area. So I can immediately see how well my model is doing with that method. So I can try that out. The other thing I can do is, if I right click on this box here, I can save this model. I can say load a model. I can visualize the errors. I've got a cost curve. I've got rock curves. I can do a lot of other things with the result from that. So it's quite a simple interface to use once you've seen how to do it. So that's the most basic form. What I can also do in here is if I click on select attributes, what select attributes does is it allows me to iteratively work through the data and find the best subset of inputs. It's this parameterization that we can use. So what I'm going to do, I'm going to choose the attribute evaluator and I'm going to check the wrapper subset evaluation. So a wrapper method, it basically uses the performance of an algorithm to select parameters. So we have two forms. Effectively we have a filter method which looks at the performance of individual features and selects the best ones from that. And then we have a wrapper method which uses an algorithm performance. What we're essentially using here is a combination of a filter and a wrapper. So we use the algorithm performance in the filter to select the best parameters. So what it does is we can use, for example, if I click choose here, we can use a random forest as the basis for our parameter selection. And then we can choose the search method. So this is various search methods. Best first looks at simple consecutive non-improving nodes, as it says. We've got a greedy stepwise, and that's a bit more aggressive in what it selects. And then we have a simple ranking tool as well. Yes. So I set, as I did before, my class. I can put cross-validation into this as well. So I can use cross-validation to check how it's doing over multiple views of the data. And I'm not going to do that because it takes more time. But if I click start, it's run through it already. So for a random forest, it's put 6 attributes forward, and then okay, did it work? It didn't work for that, so I'm just going to choose a different one that I know works, which is a multi-layer perceptron. Click OK, click start. So when we know it's running, we can see the Weka moving at the bottom there. It's put 6 inputs forward. It's predicting disease versus healthy. So it's taking a little bit of time because it's doing thousands and thousands of calculations. And if you can see, it's now identified the 4 best attributes as TNF alpha, IgG, ER, MMP9. So it's searched through all those combinations, and those are the 4 best ones that it's found. So if you've got machine learning data, if you've got proteomics data, you might want to filter it a bit first, but if you wanted to select, say, from 200 or 300 features to find the best combination, you could use that on that data to see which is the best combination of things that predicts the answer to a question. Using whatever algorithm you need to use. It's not having a good time today. So I could then go in and rerun this algorithm with just TNF alpha, IgG, ER and MMP9, and it would hopefully improve the performance. So we can use this as a parameterization tool. We've got a huge amount of flexibility in the algorithms we use. We've got a fair degree of flexibility in the search method we use as well. So that is one way of analysing our data. I'm going to show you some more that you can use with Weka. The next one I'll just show you is AutoWeka. So here what we do is we're literally setting up an experiment and telling it what to look for. So this tells us how many things to look through, batch size, how much memory to use, the number of best configurations. So this is just looking for the top single best configuration out of them all. But maybe we don't want to just rely on one. We might want to select 5 or 10. Give us the top 10 rather than just the top 1. The seed is a random number seed. So basically, if we run this with the same random number seed for the same data for the same time, we'll probably get a very similar or the same answer. The random number seed is how it's randomized, that we can fix the way it's randomized by using that particular seed. Change the seed, you'll get a different set of results. Because it's a randomized process, the way the cases are selected, the way the weights are initialized, there's a lot of randomness in there about setting up. But that can be an advantage. So if we find things that are there despite that randomness, then they're more powerful. Because ultimately it's starting off with a random state and then finding the correct state. And if we keep finding that same correct state, then it's going to be reinforced. So I'm going to not run this for 15 minutes. I'll run it for 5. And if I click OK, I want to use the same output. And then I just click Start. And as with all best machine learning algorithms, not much happens on the screen because you don't want it to, because it's a waste of resources. But working in the background, that's telling me it is. This is now looking at a whole load of different algorithms, different solutions. So this is a good time to actually run it while they're fixing the microphone. So this will just keep running. It tries lots of algorithms, lots of parameters, and it will find the best solution for this data set. And sometimes that's all you need to do. You've got a data set, you want to find the best method to use, and maybe then you can use that as a basis for further data sets that you evaluate. So that's helpful. I can't really show you how to do it because I'm doing it on the machine now. I don't want to change it, but you can tweak the settings of Java. It's not done in Weka, to increase the memory limit of Weka as well. I think it currently uses 16 gigabytes of memory, but you can change that to, if you've got a really powerful machine, 64 gigabytes of memory. That gives you the potential to load pretty large data sets into memory and let Weka run through. Now you can also tweak Java and Weka. I don't know how to do this because I've not needed to, but you can tweak it to run on a GPU. Okay, thank you. So if you tweak it to run on a GPU, you're going to get a speed up of performance because it's parallelizing it. as well. So where have we got to? We've run 98 different models so far, and we've got about 23% error is the current error rate. That's pretty bad actually. Well, it's a pretty rubbish data set. I made it for this purpose and wanted it to be noisy. So it's running, we're finding results. We can come back in a little bit and see what we're getting. Right, while we're doing that, I just want to show you another way of doing this. So in this folder are files which are part of a bigger file. We recently had a paper published on muscle aging, and this is data from that muscle aging study. But what I've done is the data structure would normally be, we have the genes across the top and the cases down the side. And we might have 20,000 genes along the top and 100 cases, 200 cases down the side. Because Weka can't really cope with that in its current form, what I've done is I've basically taken the column header, I've taken the gene expression value through the population for a particular gene, and then I've taken the state to whether it's young or old. Well, in fact, I think for this I use regression. So it's the absolute age of the person where the muscle biopsy was taken. So by splitting the data into one file per gene, that means it's more efficient. It's not loading the whole data set into memory. It's just running one data set at a time. So if you have any command of R, it's actually quite easy to split that up. You could, if you have plenty of time on your hands, you could split it up manually. But by getting R to literally extract the columns into files, you could probably do that with 3 lines of code. You could probably get ChatGPT even to write you the code to do that. But by splitting it up, you can use Weka as a way of looking through transcriptomic or proteomic data for enrichment. You want frequently the first time to enrich for a set of genes that stably answer your question. So if something predicts something, then that's interesting. So if this gene predicts muscle age, that's interesting. But in the genome, in all likelihood, there are multiple things that are going to be associated with muscle aging. And what I want to do is find from across the whole genome those things that are associated with muscle aging in a stable way. So if I look at a different randomization of the data, I will find the same gene in the top 100, 200, 300 genes. If I repeat it again, by looking at the data and coming back to the same set of genes, I will, if the same things keep coming out, I know they're important. And I know they're unlikely to be false discoveries. So this particular workflow is centered around that. And once the other part of Weka is finished, I'll show you how to use it to enrich. So, what these individual files look like is this. So it's literally a column called case. That's the individual in the population. Within that we can randomize as well, and we will. We've got the expression, and the header of that column is the gene name. And this is using GI notation. And then we've got the column header for age of the person who gave the muscle sample. And they range between individuals who are 20 and individuals who are, the oldest one I think is 84. So this is what we were doing in this study, understanding the impact of age on muscle, and particularly identifying risk factors for sarcopenia, which is what muscle wastage in old age. So I can use this data, and you see what I've done here is each one of these in this folder is a separate gene, and I can run through these and compare how they perform in an analysis workflow. Okay, any questions on what I've shown you so far? Hopefully it's being videoed, hopefully you'll be able to hear me on the video. Make notes while you're going on, take pictures, take videos if you want to for your own use, I don't mind. So, well, okay, our Weka Explorer has finished. It's run for 5 minutes. The rock area for this combination, so what did it find? Well, let's have a look. It found that in this case the best classifier was a classifier called a rule-based classifier called a JRIP. It's got the parameters for that classifier here. The error rate here, and then I can actually, if I wanted to instantiate that algorithm with those parameters into Java, I can use that string of code there. So that will set that up, as long as the right libraries are in place in Java, and run that same algorithm. So if I could put it into an algorithm for somebody else to use. So that's that bit. It gives me the usual metrics, classifier rate, it gives me correct cases, 215 correct, 47 incorrect. So that is an improvement on what we had before. It also has a confusion matrix. And let me explain a little bit about confusion matrices. They're used for classifiers, and they tell me how many are right and how many are wrong. But it's a bit more than that. So we know A is positive, B is negative. Let's move the cursor out of the way. So of the positive cases, 35 were predicted as A, and 37 were predicted as B. So that immediately tells me my algorithm is not so good at predicting positive cases. It's got more wrong than right for the positive cases. If I look at the negative cases, it predicted 180 as B, and it predicted 10 as A. So it only got 10 of the negatives wrong. So that immediately suggests there is a bias to my results. And I deliberately put that bias in because I wanted to talk to you about balance and bias in classifiers. So for example, if you look at the number of cases, well, I've got 190 negatives and 72 positives. That means the negatives are much more represented in my data set than the positives, which means that the algorithm is going to encounter negatives a lot more of the time. So what I have to do is, when I build a classifier, I have to balance the numbers, ideally. I can probably get away with 2 times versus 1 time, say 2 times A, 1 times B. But much more than that, I start to introduce bias. And I've had people who come to me to analyze data where they've got 99 of group 1 and 1 of group 2, and I just say you're not going to get anywhere with that because you need a good representation across both of the classes for the algorithm to work well. So that's an important consideration, balancing the numbers. And this is not a balanced study. If you're working on continuous values, you don't have to worry about balancing them because you have a continuous range. That's okay. So it just says if I want to get a better performance, I run it for longer. Well, I kind of knew that. But I would say to get a better performance, I should balance the numbers and run it for longer. Any questions about any of that? Okay. So the other thing I can do, I just want to show this. This is specifically for image analysis using deep learning. So what I can do here is if I label images with a class name in a folder. So let's say I have, one of the most weird ones I've seen is people who are trying to distinguish between a fajita and a chihuahua. So they've got these fajitas, which is like a flatbread rolled up with cheese in Mexican food, and they have a chihuahua, which is a small dog. And what they did is if we were to run that, then we'd have a series of images which were called fajita 1, 2, 3, 4, 5, 6, 7, 8, 9, and we'd have a series of images called chihuahua 1, 2, 3, 4, 5, 6, 7, 8, 9. And we would put them into a directory, and then we could, where it says open image, we could utilize that to pull in those images, and it would automatically convert them into arrays of pixels. So it converts them into something the algorithm can use. What I can also do is I can use the GPU for this. I just have to do a bit of Java setup. It tells you how to do that. I haven't actually set that up on my machine because it messes with another program I use. It will show, once you've loaded the images, it will show them in here. And then I can run. So I've got various algorithms, but the only one I can run in this is deep learning for J, and it's a convolutional neural network, a CNN. So this is previously set up a default convolutional neural network in here for me. I basically have pre-trained zoo models, which is it's set up some weight settings that are good for image analysis. It has I've got a model output decoder, so what that will do is it'll give me the convolutional layer components and predict something. And then I can just run it. So immediately you've got access to CNNs for analysis of images, allowing you to utilize them without, they normally would code these in Python a while. So that's a useful tool. Say you're looking at cell images, say you're looking even at imaging mass spectrometry, potentially you could use CNNs on that. So that's another technique which is available in Weka, and I'm not going to run it. I'm not going to run it right now, so I seem to be getting a lot of emails through today. So okay, that's the most simple interface for Weka. That's Explorer. I recommend you have a look at the videos, that you have a look at how to use it. You have a lot of help, a lot of support. If you want to try something, you can just Google, how do I do this in Weka? And I'm sure there's probably a video for that, or some text for that. So I'm now going to get back onto looking at that data set that I mentioned to you. So instead of using Explorer, we use for that Experimenter. So Explorer is very much about you learning to use the algorithm, trying things out in a small scale. Experimenter is about running multiple algorithms for multiple data sets. So you can run Experimenter on 20 different data sets with 20 different algorithms and find the best results. It's a way of automating the whole process. It will allow you to do things like trying out 2 different algorithms on, let's say, like the files I have, 20 different files of mass spec mass spec values, answering a particular question. It will allow you to evaluate those algorithms on each one of those features, and it will give you the results in an Excel spreadsheet at the end of it. So you can kick out the results as a report, which is in a spreadsheet. Let me show you how to do that. So in the setup phase, I can open a previously set up experiment. I'm not going to do that now. I'm going to create a new one. So the first thing I'm going to do is put the results into a file. I'm going to call that file GAN results, and I'm going to set that to go to my computer. Right, we have a GAN course folder here. Oops. So I've set up where the results need to go. I'm going to use a training test split. My problem is a regression one because age is a continuous value. I'm going to just do 2 repeats, otherwise we'll be here for a very long time. And I'm going to set data sets first. So basically it runs the same algorithm on a particular data set again and again and again. Now when it comes to adding data sets, what I can do is go to my folder where my results are, and then I know they're all CSV files in here. No, they're not zipped files. And basically click the top file, press Shift to click the bottom file, and click Open. So it's now loaded all of those files into the experiment, and we'll look sequentially through those. The file name is the gene name. That's important because that's what's going to be written to the results file, so we can deconvolute that by looking at the gene name. And now what I need to do is select an algorithm to run on those. So I'm going to, for this I'm going to use an MLP. In the MLP, I can set the architecture up, and I can set it to automatically select the number of hidden nodes in the architecture. And I do that by just putting an A in here. So it'll automate that process. You can see also I can change my learning rate, my momentum. Not sure if I can change the transfer function. If I click on false, will that round? think it's not letting me change the transfer function because it's on auto build. I'm going to use an MLP, and that's all set up and ready to run. I can add notes into my experiment if I want to. So I'm just going to click on Run now and click Start. So it's now working through those data sets, each gene being a data set, evaluating those in terms of their ability to predict muscle age. And we can use the correlation coefficient and the root mean squared error as the selection parameter for that. So it's working through, it'll take a little while. So, are there any things you would like me to clarify on what you've seen so far? Have a go, have a play. You know, there's data in Weka for you to have a go with. Use that. You will learn how to do it. A lot of people use Weka and it will produce publication quality results because of the power of the algorithms that are embedded into it. So it is useful. Yeah, it's just running through now. Takes a little time. A lot of my role as a machine learning specialist or computational biologist involves me waiting for bars to move across screens and folders to fill up with files. It's when the algorithms are running, you don't see much, but the nice thing is you can automate something and let it run for a few days and then come back and harvest the results, which is quite rewarding. Okay, it's finished. So good, that wasn't too slow. So now, if I go to the GAN course, and here you can see we have a CSV file with our results in it. I'm not going to convert those for now. Well, you can see here, in this file I've got the key data set, so that's the gene name. I've got the algorithm. I've got the options set for that algorithm, version. I've got the date and time, which I think is a non-date format here. Number of training instances, there's 43 individuals. Number of testing instance 22. Now you see I've got my mean absolute error, root mean squared error, relative absolute error. What I'm really interested in is the correlation coefficient, because I want to find the things that correlate best with muscle age. So what I can do, the most simplest form is I can sort by correlation coefficient and sort largest to smallest. Click OK. So let's put a split in this. So across the different randomizations, the best gene in its own right is GI 1009 2672. You can see I have now, this one has then 1004, then 1019. Then I've got 1009 again, then I've got 1019, then I've got 1019, then I've got 1004, then I've got 1004. So you can see across the different instances, the same three genes are coming up as highly predictive of muscle age. So I put 50 in, and I can immediately see which ones are stably coming up. And in that way, by doing that, I can say, well, I can start to enrich and say these are the genes out of the 50 that consistently answer the question of muscle age. Now you can use that for mass spec data if it's in a protein name format. You can use that for expression array data. You can use that for RNA-seq data. It's a way of generating new hypotheses based upon particular data sets and particular questions. So, any other questions? That's all I'm going to cover for Weka. There's a lot more to go at. There's a lot more things you can do, but that really kind of introduces the basics and how you can utilize it for the work that you potentially can do. Right, I'm going to end my full screen, minimize that. You can, if you've got a Microsoft Azure account, you can embed machine learning with a no-code solution in Microsoft Azure. Essentially, it's very similar to what I showed you in Orange. It's a workbench type solution. And then there is another piece of software called RapidMiner. And RapidMiner is an automated way of, a bit like Weka, of looking at, with a no-code solution, looking at large data sets and pulling out features. So RapidMiner, I think the free option only has a very small number of algorithms and arguments. You can pay a nominal fee and get a lot more. But I've shown Weka and Orange because unlike Azure and RapidMiner, they're free. What else is out there related to this? Well, I mean, there's not time here to go through coding, but the two recommended systems for machine learning, I would say the primary ones are Python. So you can see there's lots of online courses on how to code machine learning in Python. The key I find with Python is you have to install and manage the environment. And for biology, I would do that with Anaconda. So you load Anaconda. Anaconda helps you set up an environment, loads all the necessary packages, and then you can install Python and it will have the necessary environment for what you want to do. You can also use R. So R is, I would say, much more biology friendly because you've got elements like bioconductor in there. Although machine learning wise, the library, you've got PyTorch in Python and you've got Keras TensorFlow in Python. But with R, Keras is a later implementation. It's not so well developed. But you can still put machine learning algorithms in Keras. So the way these things work, just to explain, you've got these libraries. You have to install and then load the libraries. And that gives you functionality that you can put into the code. So in order to do something, you load a library. And Keras is a machine learning library. So it allows you to write code that implements machine learning. There's also Tidyverse. Tidyverse is a massive library. It allows you to use functions that are very good at talking to one another. It's good at things like data manipulation, graph plotting, things like that. And then the last one is dplyr. And dplyr is very good for manipulating data arrays and spreadsheets and the like. But you know that's a And the story behind that is quite long. So have a go, but the story is quite long. You can do things in Java. I'm not so familiar with Java, but it's less entry level. But in many ways, the libraries in Java, I think, are more powerful. There are also, and this is important for what we're going to consider soon, okay. There are some very good network biology tools. Biology exists in a network. So one of the good tools is Gene Mania. With Gene Mania, you can just drop a list of genes or protein names, but if you use the gene symbol into that, set a species, and it will identify what interactions there are in those in the literature. So it's a way of looking at gene associations across networks.
Can I just?
Yeah.
So Graham is showing you some tools, right? And if I am right, Shambhu would have shown you a list of differentially expressed proteome, right, Shambhu? With Metascape, with MetaBanalyst. So your job for the day is take a tool of your choice and create and use any of the tools that you find, especially network biology tools or Orange. You interpret the same data with your point of view. You see, the good thing about multiomics data is even if the data is lying around for 5 years and you give it to 100 people, 100 people will use their own point of view and own biology and expertise to get new insights from it. So your assignment, which will of course be judged, and this is for tomorrow morning before you reach, you can drop an email. You have the email, Yarn Proteomics AI, and even the PDF that many of you are sending, please don't send me on the WhatsApp group, because I'm getting spam. You just say assignment for day 3, so then we can skip the test part. Right? So
That's a good
it'll be
idea.
good for
Yeah.
you to use this tool.
So you could use your differentially expressed genes, the values for those.
Yeah, on your mail, you can just make a small PowerPoint presentation of maybe 5 slides. That was the differentiation of gene. I think you can ask Shambhu regarding what was the biology. He might have told you what this experiment was all about. You can go back to the original paper. Shambhu, which paper was this? Experimental brain research?
Yeah.
So you can go back to that paper. It's available. You can ask me, I will share the paper with you. And then you try to interpret something new, something novel that you find with your analysis.
Yeah, so you could drop those gene names or protein. The protein names as a SwissProt name won't work. But if you get from SwissProt the gene symbol, which is the short gene name, you can drop those into any of these and find the network biology. You could, if you've got the raw data, drop them into Weka and build a classifier that separates one group from another. Yeah. So if you've got that raw data, you could build a classifier in Weka using AutoWeka, or you could use parameter selection and find what was the best subset of those DEGs in combination that predicted the class. So you can see immediately there's a lot of ways of using this to explore your data further.
So maybe on the final day we can have a presentation randomly from maybe 5 people. And we will see how the same data, 5 different eyes look at it in 5 different points of view.
Yeah, I mean we could take the top 5 people from the quiz if they really want to. Yeah. Then another one, and this is very much protein focused. So string is about protein-protein interactions. That's important for biology and for drug discovery. But if you're looking at proteins, you want to see how they interact in a network, and it's all about differential binding. It's about energetics and so on, like so forth. This is still the one I just demonstrated Weka in Orange. So.
Okay. So
But I shouldn't be long. Maybe 10 minutes, 5-10 minutes.
After 10 minutes we can have tea.
Yeah. Okay. Yeah. So Network Analyst does something similar. Cytoscape Now, Cytoscape is a tool for visualizing interaction networks. It's a very powerful tool. You can drop in a set of input values, interaction values, and target values, and then map the network in Cytoscape. So Cytoscape is a good way of visualizing networks. Intact is another interaction analysis tool from the EBI. I'm presenting multiple options because each one will have its advantages and disadvantages. We can also look at whether our genes are involved in particular pathways. Now, there's this whole pathway hypothesis in biology. We have pathways associated with things. For example, there's a cell proliferation pathway. There's a cellular senescence pathway. There's a JAK-STAT pathway, a p53 pathway. We can see whether our genes or proteins overlap with a particular pathway by dropping them into databases. So one pathway database is KEGG, which is the Kyoto Encyclopedia of Genes and Genomes. We can literally drop the gene symbols into that. It'll tell us what pathways are coming up. More powerful still is a piece of software called Reactome that looks across multiple pathways and has a very rich set of features in it. So Reactome is very powerful. We can also, if you have money, do Ingenuity, but you can use free trials. You can get a free trial. A little tip, which I shouldn't really tell you, but if you have multiple Google email addresses, you just create a free trial on the email address, go for a month, generate another email address, create a free trial with the email address. And I shouldn't say that to you, but I know people have gone for about a year doing that. And another one is Metacore, which is a similar sort of approach. I suppose the only thing is if you generate 20 or 30 emails, you're going to get spam emails from Ingenuity for a whole year on every one of those email addresses, which is probably a disadvantage. Anyway, what I'm going to be talking about in the next set of lectures is about using machine learning to discover new biology and to discover drugs. That is about how we can go from biology to small molecules and how we can select biological features that are potential drug targets. So there are some databases around that. The good one is Drug Genome Interaction Database, DGIDB. What that will tell me is I can put a gene name in or protein name in to that, and it'll tell me what current drugs there are that will target that, whether they're in clinical trial, whether they're in proper use, and what indication they're used for. So this is a really good resource for potential repurposing of drugs. So say I've got a molecule of interest, say I want to find out what small molecule hits that, I put something into DGIB, I've got my data from lung cancer, suddenly I find a drug that's been used successfully in idiopathic pulmonary fibrosis. That drug can then potentially be repurposed for lung cancer because I've found this gene to be important and I've found an existing drug which could have an effect in that system. So that's really powerful. What I can also use is DrugNome. So DrugNome AI is a tool produced by AstraZeneca, drug company. And what DrugNome AI does is it gives me a score to tell me how druggable a particular molecule is. In biology, there are various molecules that have lots of functions. And in some cases, certain things are not very easily druggable. So transcription factors, for example, are not easily druggable. But things like G-coupled protein receptors, transmembrane molecules, anything that's kind of an enzyme is very druggable. So with Drug Gnome AI, we can actually start to drop a list of genes in, and we can say, how druggable is this molecule? If we want to get a bit more detailed, we can look at something called Chembl. So Chembl is a database of chemicals. It has proteins in there, but it has thousands and thousands and thousands of small molecules. I can put a gene name in there, and I can see whether there are any known drugs that target that. I can get their IC50, which is the concentration at which it would have an effect to kill 50% of the cells. So that is important. And so I can look at that for chemical matching. And I can also look at open targets. So what open targets has is a database which links drugs to diseases, and has a database that links drugs to genes. So open targets is another source for me to find out what small molecules there are out there that will target a particular piece of biology. So you can see how much value there is here. It gives us the potential to create a pipeline for in silico drug discovery. And bear in mind, some of the deals that go for well-validated, well-characterized drugs are in the region of $500 million. There's extensive value here. That's the end of this bit. It's time for tea. Please feel free to ask me any questions over tea. Thank you.